morning everyone. I am Sadhana, Assistant Professor from Biviraja Institute of Technology. So on today's lecture, I would like to explain about the factors affecting on drug metabolism. What is drug metabolism? So on previous classes, we already discussed about drug metabolism. When drug is entered into the body, so it will go to blood through absorption and it will distribute it throughout the body and it will show the mechanism of action. After that, it is entered into the liver for the biotransformation. So that biotransformation is also called as drug metabolism. So which is inactivated the drug. So here in this class, we have to discuss about different factors affecting on drug metabolism. So as we already know, various physicochemical factors affect the absorption, distribution of the drug. So as like that, same factors like drug solubility, dissolution rate, lipophilicity, pH, pKa, various factors affect on drug metabolism. On today's class, we'll discuss about the chemical and biological factors which affects the drug metabolism. So in chemical factors, enzyme induction and enzyme inhibition and environmental chemical, how these factors affect the drug metabolism, and biological factors like age, diet, sex difference, species difference, strain difference and altered physiology. How all these factors affect the drug metabolism, we'll see. So first, induction and inhibition of drug metabolism enzymes. So the state of enzymatic system involved in the biotransformation represents the important factors in pharmacokinetic and pharmacodynamic activity. So if the drug would like to show the pharmacokinetic and pharmacodynamic activity, the main reason is the enzymatic systems. The change in the enzymatic system is qualitative as well as quantitative. Means it affects the concentration of enzyme in quantitative, it affects the different enzyme functions in qualitative manner. So in qualitative changes are commonly due to the impairments in the state of enzymatic systems. So as we already said, any nutritional deficiency or any genetic factors which affects the enzymatic system shows the impairment of enzyme activity. And the quantitative changes mainly involves the stimulation of enzyme activity, which is nothing but enzyme induction. Mean to say it increases the enzyme activity. So next, the reduction in the enzyme activity, which also called it as enzyme inhibition, means by any external factors, xenobiotics or any other compounds and metabolites which inhibits the enzymatic activity. So the pharmacological sequences of this both phenomenon of qualitative and quantitative are modification of intensity and duration of the pharmacological action. So whether the enzyme inhibited or induced, there is a change in the pharmacological activity of the drug molecule and modification of T-half and biotransformation rate. So for example, enzyme induction, mean to say increase in the enzyme activity which reduces the half-life of the drug because the drug metabolized very fastly compared to the normal metabolism. Enzyme inhibition, which increases the T-half of the drug because there is impaired enzyme activity. So there is no proper amount of enzyme activity for the metabolism of drugs. So the drug will stay a long time in our body in an active form. Next, appearance of adverse reactions and overdosing and therapeutic efficiency. So all these three effects will produce while enzyme induction and enzyme inhibition. So induction of drug metabolizing enzymes. So what is enzyme induction? So induction is defined as increased enzymatic activity associated with increase in the intracellular enzyme concentration. So when in the body the intracellular concentration of enzyme is more, there is an increase in enzymatic activity. For genetic point of view, if we compare the genetic point of view, increase in the enzyme protein usually causes the increase in transcription of associated gene. So, in the genetic point of view, the gene transcriptase which associated with high enzymatic high enzymatic protein leads to the induction of metabolizing enzymes, which means the increase in activity of the enzyme system. So the stimulation of enzyme activity represents increase in the concentration of specific enzyme due to increase in the rate of synthesis. So in the genetic point of view, due to the gene modifications, there is an increase in enzyme protein. But in stimulation point of view, there is increase in the rate of synthesis of the enzyme. So these two leads to the enzyme induction. So the direct consequences is accelerated and increased biotransformation of the drugs, endogenous compounds and xenobiotics by co-administration with any other compound designated as inducer. If we'll give the drug with any other compound which increases the activity of the metabolizing enzymes, we called it as enzyme inducer. So this inducer have the pharmacological activity of the drugs, the increase in the metabolism. So which the drug is involved in the specific interaction, and decrease in the quantity of drug available for the pharmacological activity. So by this enzyme induction, the drug metabolism increases, the drug activity decreases because of there is not efficient concentration of the active form of the drug is available in our body. So there is a decrease in concentration of the active form of the drug available in the body. So that's why there is an impaired pharmacological activity of the drug in the body. So what are the mechanisms of enzyme induction? So which type of mechanisms acts for induction of an enzyme? So first one, increased both liver size and liver blood flow. So if the liver size was increased or the blood flow through the liver system is increases, which increase or induce the enzyme activity. So increase in both total and microsomal protein content. So what are the proteins which are affect the synthesis of this metabolizing enzymes? If the concentration of the protein increases, there is an increase in concentration of the enzymes. Next, increased stability of enzymes. So after some time, the enzymes were degraded because of their lifespan. So if the stability of enzymes increases, which also induce the metabolism. Next, increased stability of cytochrome P450. So we already discussed this cytochrome P450 is a metabolizing enzyme which mainly acts in phase 1 metabolism. So if this stability of the cytochrome P450 increased, there is an increase in the drug metabolism. Decreased degradation of cytochrome P450. So increased the stability, decreased metabolism. So if the concentration of enzyme increases or the degradation of the enzyme decreases, also affects the enzyme induction. Next, proliferation of smooth endoplasmic reticulum. As we already discussed, the non-microsomal and microsomal enzymes come from mitochondria, cell cytoplasm and smooth endoplasmic reticulum in the cell. So if the proliferation of smooth endoplasmic reticulum increases, there is increase in the concentration of cytochrome P450 and all microsomal enzymes, which leads to induction of drug metabolism. So the consequences are, so if the drug metabolism increases, there is no active form of the drug which is available in our blood to show the pharmacological activity. So thereby the pharmacological activity of the drug decreases, and increase the activity of the metabolites if the metabolites are active. So this is used in case of the prodrugs. If the drug is initially in active form and later on after metabolism if it is converted into active form, this enzyme induction very useful for producing active metabolite for mechanism of action. Next, altered physiology due to the enhanced metabolism of the endogenous compounds such as sex hormones. So it also affects our body as physiological status because of the increased metabolism of the sex hormones. So we have two different types of the enzyme inductions. So it is which is substrate dependent. First one includes induction of phenobarbital. Influence of phenobarbital of the metabolism and duration of action of different type of drugs. If we take phenobarbital with any other drug, or pre-treatment of phenobarbital to the concerned patient with, if you would like to take another drug, influences the drug metabolism. See here the examples. The oral anticoagulants. The anticoagulants effect decreased due to the increase in metabolism. So if we take phenobarbital with oral anticoagulants, mean to say, so which prevents the coagulation of the blood. So the activity of the anticoagulants decreases due to the increase in drug metabolism. Tricyclic antidepressants, mean to say the antidepressant drugs which decreased the effect of depression because of the increase in metabolism. So same like corticosteroids, narcotics, theophylline, muscle relaxants, all these drugs affected by phenobarbital. If we take these drugs with phenobarbital or pre-treatment of phenobarbital after taking these drugs also shows the enzyme induction which reduces the mechanism of activity of the specific drugs. Next type of enzyme induction is polycyclic hydrocarbon type inducers such as 3-methyl-chloranthrin and cigarette smoking, which stimulates the metabolism of different varieties of drugs. So that's why if we visit the doctor, initially he will check for any habits which associated with the concerned person, as like cigarette smoking and alcohol ingestion, everything. So because this cigarette smoking and taking alcohol and some type of drugs combination with our drugs also induced drug metabolism. So drugs induced metabolism due to cigarette smoking have the clinical consequence like theophylline, caffeine, tacrine, imipramine, haloperidol, pentagocine, propranolol, flacidin and estradiol. All these type of drugs shows different type of clinical consequences with cigarette smoking. Big means if we are having a habit of cigarette smoking and we are taking all these drugs simultaneously with cigarette smoking, by induced drug metabolism, these drugs lost the activity due to the higher metabolism. Some drugs such as carbamazepine, meprobamate, cyclophosphamide and rifampicin. So these type of drugs are stimulate their own metabolism. Mean to say that specific drugs only increases the enzymatic action of the enzymes. So that's why there is increase in metabolism of the drugs. So we call it as auto-induction or self-induction. Mean to say that same type of drugs increases the mechanism of activity and reduces the pharmacological activity. That's why we call it as self-inducers or auto-inducers. So you see here the inducers, drug enhanced metabolism, barbiturates, alcohols, phenytoin, rifampicin and cigarette smoking. So all these type of drugs, if we combine with the specific drugs which increases the enzyme activity, thereby reducing the pharmacological activity of the specific drug molecules. So this is about the enzyme induction. So next we'll go to the enzyme inhibition. So it is opposite to the enzyme induction. In enzyme induction, we'll discuss the increase in metabolic activity of the enzymes because of increase in enzyme concentration or increase, mean to say qualitatively and quantitatively. So here enzyme inhibition by pre or co-administration of some type of drugs with xenobiotics as a well-recognized phenomenon with pharmacological and toxicological activities. So if we take different type of combinations of drug, in that any drug inhibits the enzyme activity which reduces the metabolism and increasing the pharmacological activity as well as toxicological activity of the drugs because of the high concentration of active form of drug present in the body. So this inhibition takes place in different pathways. The mechanisms like blocking enzymatic synthesis. So if any drug will block the enzymatic synthesis, which is reduces the enzyme activity. Next, destruction of preformed enzymes, the enzymes which are responsible to produce specific microsomal, non-microsomal enzymes which are responsible for the drug metabolism. If that enzyme destruction happens, it also inhibits the drug metabolism. Inactivation of enzyme by their complexation of drug metabolites. The same drug metabolized and the metabolites will inhibit the concentration of enzyme. So it is also a possibility of inhibition of drug metabolism. The consequences are delayed in biotransformation of certain drugs, resulting in increased plasma concentration. So we already discussed, if the enzyme activity was reduced, means what happened? There is no enzymes, or required amount of enzymes are not available for the drug metabolism. Thereby, high concentration of drug is present in the body, which we call it as increased plasma concentration. Next, prolongation of their pharmacological action. So it also prolongs the drug activity because of high amount of active drug is present in the plasma. So basic mechanisms of enzyme inhibition involves two types, which is direct inhibition and another one is indirect inhibition. In direct inhibition we have three classes. One is competitive and another one is non-competitive and product inhibition. So in indirect inhibition, repression and altered physiology. All these factors affects the enzyme inhibition. So first one, direct inhibition, competitive inhibition. So any two substrates simultaneously will go to the enzyme to compete, so thereby reduced enzyme activity, thereby inhibition of proper amount of enzyme. So results when the structurally similar compounds compete for same active site in the enzyme. Next, such inhibition due to the substrate competition. So this is due to substrate concentration. If which drug we would like to increase the reduce the pharmacological activity, so we can increase the concentration of the substrate. Thereby the competitive inhibition happens. Example, methacholine inhibits the metabolism of acetylcholine by competing with cholinesterase. Cholinesterase is an enzyme which is used for the metabolism of acetylcholine. So if we take the drug methacholine in combination with acetylcholine, this methacholine competes with the enzyme and inhibits the metabolism of the acetylcholine. So in non-competitive inhibition, there is not structural similarity of the compounds, but the other substrate which is attached to another site of the enzyme and inhibits the binding of the specific drug to the same enzyme. So that is called non-competitive inhibition, since the interaction is not structure specific. Example, metals like lead, mercury, arsenic and organophosphate insecticides inhibit the enzymes non-competitively. So that's why this metal poisoning happens, because of reduced enzyme activity. Next, product inhibition. So it results when the substrate combined with the enzyme, it produces the product. So this product again it competes with the substrate with the same enzyme. We call it as product inhibition. So indirect inhibition, first one is repression, means the reducing the synthesis of the enzymes or decreasing the enzyme content. Next, altered physiology due to any nutritional deficiency or hormonal deficiency. There is due to the nutritional deficiency and hormonal imbalance. So there is inhibition of drug metabolism. So next, different factors like biological factors, age, diet, sex difference and species difference and strain difference and altered physiology. So in age, different ages. Say for example, see here, the ability of the microsomal enzyme metabolize the drug poor in immature implants compared to the adults. So the drug which is caffeine have half-life of 4 days in neonates, mean to say newly born babies. In adults 4 hours. So this is based on the age difference, and the conjugation with sulfate is well developed in newborns compared to the glucuronidation. That's why the high amount of sulfate metabolite of the paracetamol was released in the body of the kid, so which is converted as hyperbilirubinemia. And similarly, the sulfonamides cause renal toxicity and paracetamol cause hepatotoxicity because of premature this glucuronidation enzymes or improper development of the liver enzymes. And the children between 1 year to 12 years has high metabolic activity compared to the adults. That's why for the kids, we are giving the high concentration of drug mg per kg compared to adults. So next, diet. Low protein diet decreases and high protein diet increases the metabolizing ability. And the protein carbohydrate ratio, if the protein carbohydrate ratio is high, the microsomal mixed function oxidase activity is more. So fat-free diet reduces cytochrome P450 levels. This is very important compound for the microsomes. Next, dietary deficiency is also highly effective in the drug metabolism. So grapefruit inhibits the metabolism of different varieties of drugs. So thereby, if we take any drugs with grapefruit, the concentration of the drug is more and the activity also more. Next, starvation also reduces the glucuronides. That's why the patients with diabetics has reduced glucuronide because of drugs there is high amount of glucose was not available for the synthesis of glucuronides. Next, malnutrition in women results in the enhanced metabolism of sex hormones. Alcohol ingestion results in the short-term decrease followed by increasing the metabolic activity. So next, sex difference. The women metabolize the benzodiazepines slowly compared to the men. Studies shows that women with contraceptive pills metabolize a number of drugs at slow rate because of high amount of estrogen circulates in the woman's blood. So species difference. So here different species. See here in men and in rabbit. So the metabolism of amphetamine and ephedrine is predominantly metabolized by oxidative deamination. So, in men and rabbit, this amphetamine and ephedrine is metabolized by oxidative deamination. But whereas in rats, the aromatic oxidation is the major route. So, there is two different metabolizing systems for the same drugs in the different species. So, another example, in pigs, the phenol is excreted mainly glucuronide. So, in pigs, the glucuronide conjugation happens for the phenol, but sulfate conjugation happens in the cats. So in pigs, the same phenol is excreted through glucuronides and the same phenol is metabolized through sulfate conjugation in cats. So that they're like different species have different enzymatic systems. So there is different drug metabolism for the species. Next, strain difference. Strain difference means different species. Say for example, see here. So different groups like whites, blacks, Latin Americans, Indians, and Japanese and Eskimos. So N-acetylation of isoniazide. So isoniazide is a drug for the metabolism of isoniazide. This N-acetylation is a pathway. Slow acylators, see here, slow and rapid. In whites, US and Canada, 45 and 55. In the blacks, 48. In Latin America, 67. In American Indians, 79. In Japanese, 87. In Eskimos, 95. So there is slow acetylation reaction happens this much of percentage in different species. So which states that the metabolism depends on the species difference also or strain difference also. Next, altered physiology, pregnancy. So, maternal drug metabolism ability in animals is reduced during the later stages of pregnancy. So, if pregnancy going on week by week, so the phase 1 and phase 2 reactions will slow down because of the high sexual hormonal enzymes like estrogens were circulated in the body. So this suggested the high levels of steroid hormones circulating during the pregnancy. So as we already discussed, high steroidal hormones like estrogens circulated throughout the body. That's why there is reducing the metabolism during pregnancy. And see here, the anticonvulsants. The hepatic metabolism of the anticonvulsants is high in induction of drug metabolism enzymes circulating of progesterone. So some of the hormones will reduce the drug metabolism. Some of the hormones like progesterone increases the drug metabolism during pregnancy. Thank you.
Like, share and subscribe. Hit the bell icon for more updates.